Hi guys, TA Zach here, and this will be the first of a series of lecture summary videos that you can use for your labs, exams, and just getting a better understanding of exercise physiology and the things that we go over in lecture. So for week one, we mainly started on bioenergetics and metabolism, and if you look back on those PowerPoints, you'll see the key points to review. That's exactly what I'm going to go over throughout this video. So the very first thing that you probably want to understand with exercise physiology is bioenergetics, what it is, how it works, and why it is so important. It's so important because it's the basis of life. And it's the basis of life because nearly everything in the body runs on ATP, adenosine triphosphate. There's not a single mechanism in the body that is not mediated by this molecule's breakdown. How we know that ATP is that important molecule and how we get to that point is through the law of energy conservation and thermodynamics. So law of energy conservation, essentially that energy is not created or destroyed. It's merely just transferred. It can change forms, it can change into the environment, into our bodies, into plants, etc. But at no point can we just take energy out of our atmosphere or try to create energy and put it into our atmosphere. It's all transferred in some way. So thinking about that from a human perspective, we take in energy by eating. When we move, talk, exercise, whatever it may be, we expel that energy back into the atmosphere. With thermodynamics, this is our quantitative look in how we bring in energy. So thermodynamics, mainly looking at things from a kilocalorie perspective. If you've heard that word before, read it on a nutrition label. All our food is broken down into calories so that we can somewhat quantify the amount of energy that we're taking in, and then through various methods, we can almost figure out how many calories that we are expelling back into the environment. So now that we have a better understanding of bioenergetics, we have to now understand how do we get the fuel to make our energy. So that comes from substrates, and if you see I've written it in big caps, fuel with quotations, but that's just the food that we take in, and then how we reutilize it and form it back into a usable source that we can use to produce ATP. First one here on the list, glucose or glycogen, which is derived from carbohydrates, and that will give you around 4 calories per gram, so looking at it from that thermodynamic perspective, per gram of carbohydrates that you eat, it yields about 4 calories of energy. The next one on the list, fatty acids, is derived from carbs or fats, and it depends based on the internal environment and the demands of that day and when you eat those types of things. But once stored into a fat, it's around 9 calories per gram. So you see a bigger bang for your buck there, which will also, you'll also be able to understand later why oxidative phosphorylation or aerobic metabolism yields a greater amount of ATP compared to glycolysis. Last on the list, you have amino acids, which are derived from protein, and that's also about 4 calories per gram, similar to carbohydrates. The issue with amino acids is that makes up a lot of your structures, such as muscles, tendons, ligaments, and most of your connective tissue. So you don't want to use this as energy because it's almost breaking down your structures that you're trying to build up most of the time. So now that we've discussed substrates, we can talk about catabolism versus anabolism, essentially breaking down versus building up. You'll sometimes hear people talking about how they're going catabolic because they haven't eaten in like 2 hours. Not exactly the case, but that's a discussion for another day. An easy way to understand when something is catabolic versus anabolic is just think about exercise or eating. With exercise, it's a highly catabolic activity. You're breathing heavy, there's a lot of ATP demands, you've got to break down glucose and fatty acids to make energy and ATP. And then with eating, you're intaking these carbohydrates, fats, proteins, and then converting them into the substrates that you can then use later for those catabolic processes. And an easy way to know if something is catabolic or anabolic is just to look at the suffix. If it's lysis or lysis, such as glycolysis, lipolysis, proteolysis, that substrate is being broken down for energy. On the other hand, if the suffix is genesis, so glycogenesis, lipogenesis, or proteogenesis, that substrate is being used to make a storage of energy, and then now Let's dive a little bit deeper into how energy is exactly broken down so that we can use it. And this is from the ATP hydrolysis reaction. As I just said in the last section, hydrolysis, this is a breakdown or a catabolic reaction. If you see on the reactant end, you have ATP and water. That's where you get the hydro. These are the reactants. When these two combine, they are then broken down here through the enzyme ATPase. Once these two molecules are combined and ATPase is activated, you'll get the end products right here of ADP, adenosine diphosphate, inorganic phosphate, one of them, as you can see, from the triphosphate now to a diphosphate. So this is free. You get a hydrogen atom from water, and you get heat. All of these here are going to be a reason for exercise fatigue. So the more you have to do this reaction, the more likely it is that your intensity of exercise is at a level that's going to cause fatigue. The lower the demand of the exercise, the less ATP you're going to need to sustain performance. So next on the list is the location of energy systems. You can see here, this is my cell drawing. There is just cytosol and a mitochondria for the purposes of this explanation. Know that there are more things in a cell than that. This is just to enhance your understanding of energy systems. Anaerobic is going to take place in the cytosol because no oxygen is needed. So this is your phosphocreatine and your glycolytic system. Your aerobic is going to happen within the mitochondria, which is your Krebs and electron transport, and this is because oxygen is essential for these to take place. Because anaerobic systems do not need oxygen, they do not need to take place in an area that doesn't utilize oxygen. So at this point in the video, I'm going to start getting into the specifics of each energy system, starting with your most immediate, well, not most immediate, you do have free ATP just in your cell, but that's about 1 to 2 seconds. Didn't feel like that needed a section of the video, just know that. The next most immediate is your phosphocreatine or your ATP PCR system. Here's the chemical equation, so ADP plus PCR, phosphocreatine plus a hydrogen, mediated through the activity of creatine kinase is going to give you ATP and free creatine. It's important to understand here that this reaction is resynthesizing ATP from ADP. Also notice that the phosphate binds to the creatine, so by making this ATP, you get a free creatine, which means it can go back and then bind to another phosphate so that it can restart this reaction again. So you can have rapid resynthesis of ATP for energy. The only issue with this is that it is for very short durations, about under 12 seconds. This could be considered like your 100-meter sprint, weightlifting, shot put, very powerful, very quick movements. Once you get outside this time limit for a given intensity, you're not going to be able to synthesize ATP fast enough for the demands of that exercise, so we're going to have to move into a higher level of energy production. Also know that creatine kinase, the enzyme here, is the rate limiter, and this is due to ATP and ADP ratio. When energy demands are high and you're breaking down a lot of ATP, you're going to make a lot of ADP. ADP is going to signal through your body, through feedback, that creatine kinase needs to turn on and start making ATP before you die. On the other hand, if ATP and ADP are in a reverse ratio where ATP is high and there's not a lot of broken down ADP, this enzyme is going to be inhibited and we're not going to have a heavy contribution from this energy system. So now, moving to the next immediate energy source possible, we're going to talk about glycolysis or glycogenolysis, my personal favorite energy system. Once again, going back to a little bit earlier in the video, lysis, breakdown of glucose or glycogen. Look at how that flowed. It's like I thought of it or something. Understanding this is just understanding like a very minute detail about ATP production, but it does matter. So glucose versus glycogen, all it does is changes the set point for reactions. When glucose is the first molecule coming in, it has to go through the reaction with hexokinase, which is going to cause a ATP to ADP cost. So you're going to have to use an ATP to make glucose into the next molecule that it can go through glycolysis. We're also going to discuss this in the next section. I'm going to draw it out a little bit. This is just more of the wordy stuff that doesn't look like the actual process itself. With glycogen, it can bypass that hexokinase reaction, and it can become that byproduct immediately, so you pretty much save an ATP that will get you back later down at the end of glycolysis. Next thing to understand with glycolysis is GLUT4 translocation. These are found in muscle, adipocytes, and cardiac tissue. They're insulin sensitive, so that means when you eat carbs, you're going to release insulin, which we'll discuss later down the road in class. But this is going to increase the activity of these GLUT4s. And it's pretty cool. All it does is it's kind of resting in the cell, and then it translocates, so it's messaged by insulin and a mediator to move to the membrane of the sarcolemma, or in the adipocytes case, just the fat membrane. And the glucose molecule attaches to it, and then it brings it back down into the cytosol where it can do glycolysis. It's also mediated by calcium and protein kinase C. So these are very prevalent in muscle contractions, right? When we need calcium to cause a muscle contraction, something we'll also discuss later down the line in class. And all GLUT4 really does is it just increases the glucose uptake to the cells. And this is important because your ability to take glucose into the cell and utilize it is going to be beneficial for performance, but also health. Big thing with type 2 diabetics, and why exercise is recommended for them, because of their insulin resistance. When you exercise, you get an increase in the expression of these GLUT4s, so they are able to use insulin and handle carbohydrates and taking that into their body better, so that it's used and converted to glycogen, if it's not used, instead of fat. As you'll see, most type 2 diabetics are typically overweight and obese. Last thing before we go into the actual breakdown of glycolysis is the PFK, so phosphofructokinase, is the rate limiter, and this is based on the pyruvate to lactate ratio, similar to how phosphocreatine system, creatine kinase rate limiter was based off ADP to ATP, but when pyruvate is outweighing lactate, PFK is going to be inhibited so that you can preferentially use oxidative metabolism to make ATP. And in the other sense, if lactate is higher than pyruvate, PFK is going to be busting out so that it can keep up with the energy demands of the exercise. And something that might get discussed down the line, I just think it's an interesting fact that I've learned, but citrate, this is a molecule found in the Krebs cycle and oxidative metabolism. It will actually inhibit the action of PFK. So this goes back to what I just said, where when pyruvate is increased, because pyruvate, which I'll discuss next, will turn to acetyl-CoA and go into the Krebs cycle, and eventually get the citrate. It'll go back and stop PFK from doing its reaction so that Krebs can continue and glycolysis can shut down a little bit. So now we're going to go over kind of how glycolysis looks and certain steps that are going to heavily impact the outcome of glycolysis. This is an extremely simplified version. I just put the regulating enzymes and really just the end products. I didn't add any of the step-by-steps or more of the non-significant enzymes, if you want to call them that, because I think this is mainly what you're going to have to focus on for labs and exams, etc. Starting out in glycolysis, you have glucose, which is a 6-carbon molecule. That will come into play later. And then at first, you're going to have this hexokinase reaction, ATP to ATP, so we need to break down or use energy to actually get the step to occur. As I just discussed, if it's starting out as a glycogen molecule because of the enzyme glycogen phosphorylase, which I'm not sure if you went over, but just to know that, it's going to break it down into a different molecule, which can bypass this first reaction and insert itself in between the steps of hexokinase and PFK. And this is going to save you an ATP down the road. So essentially, it's more energy efficient to use glycogen as a substrate versus just straight glucose. From there, you're going to get to the PFK, so phosphofructokinase, that regulating enzyme of glycolysis. Once again, you have that hydrolysis reaction using an ATP to get this step to move along. And this is at the point where, or a little bit before, but it's going to branch off and cut that glucose molecule into 3 carbon molecules. Eventually, we get down to pyruvate kinase, once again ATP hydrolysis. It's going to become a pyruvate molecule, which is a 3 carbon molecule. From there, there's 2 possibilities. There's slow glycolysis, which is called oxidative glycolysis, and there's fast glycolysis, which is called non-oxidative glycolysis. I could say more on that topic. I'm not going to go on to a rant. I'm just going to go over the very basics of kind of enzyme kinetics and concentrations and whatnot, and just give you a simple look at it. But if you ever want to talk about that stuff, come to me. We can chat. When oxygen is sufficient, pyruvate dehydrogenase can be activated. It's a complex of proteins. Not to go any deeper than that, but it will allow pyruvate to become acetyl-CoA, go through the Krebs cycle, and eventually electron transport chain, which is your aerobic metabolism, and that will yield higher ATP. That's something we'll go over probably the next week. When oxygen is not present, lactate dehydrogenase is going to be the main enzyme that is activated, producing lactate. The thing with lactate versus acetyl-CoA is that lactate, as an end product, is really only going to give you 2 to 3 ATP as a byproduct of this energy system, whereas acetyl-CoA, once it goes through its whole cycle, gives you about 31 to 32 ATP. So it's definitely a better option for acetyl-CoA to be produced through slow glycolysis, but depending on the demands and the intensity of the exercise, lactate might preferentially be produced. And with that, on to this next session, we're going to talk about why lactate isn't actually such a bad thing and why I like it so much. Finally, onto my favorite topic in exercise physiology, we're going to discuss lactate. First things first, lactate is not an acid. Lactic acid is not a thing. It cannot exist in the body due to the biochemical structure. If it is produced, theoretically, it is dissociated very quickly into lactate itself. Why we thought it was an acid is because it is usually synonymous, lactate, it is usually synonymous with hydrogen. But hydrogen is not caused or even made, well, not made, but it's not produced from glycolysis. H+, going back to the very beginning of the video, it's from ATP hydrolysis, right? So the more we have to make energy and break it down, the more hydrogen is going to be just floating around that we're going to need to get rid of eventually. And the harder things get and the more energy and intensity goes up, or energy need and intensity goes up, the more ATP breakdown, the more hydrogen is going to be chilling. So a great part, well, before I go on, lactic acid is not because of the burning feeling. Very, very important that you understand that. Get that out of your head. It is going to kill you down the road if you just keep thinking like that. Another great thing about lactate is that it's actually oxidized in the mitochondria, and its oxidation is due to, it's a buffer, and it's a buffer for hydrogen. That's why it's seen with hydrogen, because it picks up hydrogen and then it brings it to type 1 fibers so that it can be oxidized in the mitochondria. And this is through lactate shuttle theory, where you have 2. I think Dr. O'Neill only discussed 1 in class, but I think it's important to understand how both work because science. MCT1, so monocarboxylate transporter 1, is your uptake of lactate in type 1 fibers. MCT4 actually releases lactate from type 2 fibers. Later on in the semester, we'll start going into the differences between type 1 and type 2 fibers, but just know for now in this explanation, type 1 fibers are your aerobic mitochondrial dense fibers, and your type 2 fibers are your glycolytic lacking mitochondria fibers. And I did a little graphic here. You have the type 2, which because it's glycolytic, it's going to make a lot of lactate. MCT4 is going to kick it out of the cell. It can pick up hydrogen on the way, whatever it may be. Once it kind of moves across the blood a little bit or to an adjacent fiber, MCT1 is going to allow lactate and that hydrogen to go in. Here's your mitochondria, and then it is oxidized. How this works is that the NAD, which is something that you'll find out more about in aerobic metabolism when we discuss it, needs hydrogen as a cofactor for it to work. Well, NAD itself is a cofactor. It needs hydrogen to work. So by lactate bringing in that hydrogen with it to the mitochondria, it can donate it to NAD, which then can help with aerobic metabolism. And then another great thing about lactate is the Cori cycle, which is found in your liver. If you just thought that it was for detoxification from you know what, your liver is a little bit more cool than that. Lactate is a precursor for gluconeogenesis, and what that means is gluconeogenesis is the making of new glucose, back to that discussion earlier in the video about the suffix genesis, making. By using lactate and it moves around the body in the blood, it can end up at the liver, where it's then used to make new glucose so that you can use it again for energy production. It's pretty freaking sweet. This is better because there's another thing for gluconeogenesis, your amino acids, so your muscles' proteins, and that stuff is not great for energy production because you're going to lose muscle mass that way. And so to wrap it up, we're going to talk about the pros and cons of glycolysis in general. Now looking at the pros and cons of glycolysis, as if someone was to ask you, what are the pros and cons of glycolysis? Why is it good or why is it bad? Pros, it's a rapid ATP source. It works in very short durations, pretty much 120 seconds or less, and it also depends on the intensity. It can be even shorter than that. But it's also efficient in regard to time usage. So if you're someone who's like a 400-meter runner, you probably want to get as good in glycolysis as possible because that's going to help you resynthesize ATP faster for that given demand of sport. Now, if you were a 3,200-meter runner, you probably don't want to get as glycolytic as possible because you're going to burn out within 400 meters of that race. And that rolls into the cons where it's a low ATP yield overall. If you're doing a long duration exercise that is going to require a ton of ATP production throughout, such as maybe like the Boston Marathon, you're going to need a lot more ATP than just 30 seconds rapidly. You're going to want to use various substrates and probably not your glycogen preferentially because you're going to run out of it before you finish the race, which we'll discuss later throughout the course. Also, because of increased contractions from ATP due to glycolysis and your rapid ATP demands, hydrogen concentration is going to be higher, and if lactate can't buffer that in time, then it's going to bind to troponin, which we'll discuss later as well, but that's pretty much going to inhibit muscle contraction, so you just stop. But there are also other fatigue mechanisms. This is not the only one, and that's something like, I don't know if we're going to go into it, but if you would like to talk to me about it, I'm more than happy to share. In conclusion, if you were just to skip to the end of the video, these three main things is what I'd want you to take away. Without ATP, there is no life. It is not possible. Thus, you must understand how substrates work, how ATP is broken down, how it's resynthesized, so that we continue to survive, and then even more than survive, perform at the highest levels possible. Next, Phosphocreatine System ATP PCR. Know that this is less than 12 seconds and all the related things that go into that, such as creatine kinase, the byproducts, ADP, hydrogen, inorganic phosphate, and heat, how that's going to affect exercise. And also know why creatine kinase will be active and when it will not be active. And finally, going into glycolysis, know that usually it's going to be in durations lower than 120 seconds, depending on the demands, and that also depending on these demands, we're going to have either lactate versus slow, which will be acetyl-CoA, which will end up going into more in the subsequent week. Anyways, thank you for watching. If you have any questions about the curriculum or what I discussed, please feel free to reach out to me, and have a great week.